Good morning, everybody. This is Zhiwei Chen from University of Hong Kong AIDS Institute. And I want to thank the meeting organizer for giving me this wonderful opportunity to sharing our experience for research and development using PD-1-based DNA vaccine against COVID-19. You already heard from Percy about the background of the immunocure. In recent years, we have been based in Hong Kong Science Park, and the lead products, we already have 2 PD-1-based vaccine entering or will be entering the human clinical trials. At the same time, we also build up antibody platform of technology, including the humanized antibody and also human single B-cell cloning technology. And with that, we actually have one candidate antibody called the anti-Delta 42 PD-1 antibody being under development against cancer. At the same time, we also discover very broadly neutralization antibody against the COVID, basically SARS-CoV-2 infection. So now we already have candidate 2 very potent antibody against Omicron and actually against all the variants of concerns. So this is under development. At the same time, we're also doing research to discover viral entry inhibitors derived from Chinese herb. It's a natural product and which show very broad antiviral activity against the SARS, COVID-19, and influenza, and even Ebola. So this one, we hope we can develop a new product using as a nasal spray, because a lot of vaccine-induced neutralization antibody cannot reach the nasal, that's the site of the viral entry effectively. Therefore, developing entry inhibitor is one of the very useful way to prevent the viral transmission and stop the pandemic. So this is what I'm showing. We are developing a prototype we'll bring into the, hopefully into the clinical use quickly. So talking about the core technology of platform is the PD-1-based vaccine. So this technology, I'm showing here the irregular shape of the cell is called the dendritic cell, which is the major antigen presenting cell. And Professor Ralph Steinman won Nobel Prize in 2011 for his discovery of this type of the cell. And based on Ralph Steinman's research, they use antibody to deliver antigen to dendritic cell. And in their study, they can promote the CD4 T cell response and promote the antibody production. But somehow there's no good T cell immunity induced. But using our PD-1 targeting technology, not only we can induce antibodies through the helper of CD4 T cell, we can actually engage the antigen cross-presentation for inducing very potent CD8 T cell. And people may know CD8 T cell is also called killer cells, which is the major immune cell to eliminate cancer and also to eliminate the virus-infected cells. So this is a very unique way to induce CD8 T cells. So using that currently, and actually Percy already talked a lot about the advantage of the PD-1-based vaccine. One area I want to emphasize is the PD-1-based vaccine can be repeatedly used. So this is will be very important as a booster vaccine for future clinical use against COVID-19. If we can induce very long-lasting and also very potent memory T cell responses, which can confirm long-term protection, which so far the current vaccine, we do not see such advantage at the stage. So then, as I mentioned, the 2 vaccines, one is developed for COVID for preventive use, and the other one is developed for HIV for immunotherapeutic cure use. So for the COVID vaccine, we actually making the soluble PD-1 domain infusion with SARS-CoV-2 research binding domain as the vaccine design. So the receptor binding domain is the site of the virus which will interact with the receptor on the host cells to enter the target cells. By inducing specific antibody and T-cell immunity targeting in this area, we have a better chance for either prevention and also for therapeutic use. So this is the real data we generated in the mouse model. And currently, we can see the PD-1-based vaccine, now we call the IC-COV, which can induce a higher level of the neutralization antibody when we compare with the standard no PD-1 or the inactivated vaccine in parallel. And so far, the neutralization antibody induced, and many of the animals still have neutralization antibodies against all current SARS-CoV-2 variant of concerns, including the Omicron. As we can see here, inactivated vaccine has a minimum effect for inducing such anti-Omicron antibody, but our PD-1 targeting vaccine can do so. And most importantly, the advantage is for inducing long-lasting T-cell immunity. And particularly in the animal studies, we show that the targeting vaccine through PD-1, we can induce very potent CD8 killer cells in the lungs of the animals before the viral challenge. And this advantage has not been found with the inactivated vaccine, because inactivated vaccine is not designed for inducing the long-lasting T-cell immunity for protection. So if this characteristic can be replicated in human studies, we hope our PD-1-based vaccine will be very useful as a booster vaccine for long-term protection against COVID-19, against all the variants of concern, because the variation for T-cell immune response so far is still minimum, not like the immune escape for the neutralization antibody. And after that, we also did in vivo challenge to see whether the IC-COV will induce any protection in vivo. So this is the transgenic mice expressing the human receptor ACE2, and they are very susceptible to the SARS-CoV-2 infection. And using this model, we can see the vaccine-induced immunity and protect the lung very well. This is a control animal. You see the inflammation and the damage in the lung. But in our IC-COV immunized mice, the lung actually very much fully protected. And at the same time, majority of the animal, there's no live viruses in terms of infectious unit can be detected. And also at the same time, some of the viral load already can achieve about 2 logs. That is about 100-fold reduction and in this model. And then in a different model, we also see similar challenge protective effect, suggesting the IC-COV indeed can confirm protection in 2 different animal models in vivo. So with this information and the data received, we started the human clinical trial at the Hong Kong U Clinical Trial Center. And so far, this trial is still ongoing. Hopefully in the near future, I can share some of the immunogenicity data with you. But so far, after the inoculation and the vaccine is well tolerated, there's no major side effects being reported. And then, because the DNA vaccine has a very good advantage for adapt to the variant of concern. So we can design the vaccine, and through the synthetic biology, we can generate a new type of vaccine very quickly. So far, we already made this 2nd generation PD-1-based vaccine as a booster against the Omicron. And so you can see through the parallel comparison, the inactivated vaccine cannot boost good neutralization antibodies against Omicron, but our IC-COV vaccine can do so. So suggesting the PD-1-based vaccine as a booster also has a high potential for future clinical use. So that's all I want to share with you today, and thank you for your attention.